And good morning from the US. And so today we'd like to present our platform to utilizing the Evosep to address the major questions in the TB, in the mycobacterial detections. And so here is my disclosure. And we have a few startup and working on the different technological platforms. And here is the overview for our the technologies under development in our group. And so we're starting from we're starting from the marker discovery, and no matter it's on the antigens or cell-free DNA or the extracellular vesicle, then to address the urgent need for disease diagnosis. Then we develop the method for clinical validation, and then also moving to the smart device development. But today the topic is on the Mycobacterium. This is a big family, and also it's a top strat to the human health at today's world. So they have almost like 190 the different type of species, and under each species they also have a lot of subspecies. So this is a huge family, actually containing the thousands of family members, and making our identification extremely challenging. So the PCR is pretty limited to few mycobacterial species, and always misidentify the close related the species or the subspecies. And other mass spec based technology, they need a biosafety level 3 environment, and also the large amount of the samples. So that's actually it's not practical. And also in this big family, and everybody know the oldest brother is tuberculosis, mycobacterial tuberculosis. And a few years ago, when I gave a talk on the conference and TB focused conference, a speaker comments this pathogen in this way. This is the most successful, not one of the most, the most successful pathogen in the human history. So we know the TB back to thousands of years ago, and we started understand that the TB biology hundreds of years ago. But even till today, and the diagnosis for tuberculosis is still very challenging for us. And so here is the factor for tuberculosis, and this pathogen is still infecting the 1/4 population in the world. And although the most of them are latent TB infection, and you don't have to worry about them when they're latent, but we still have like 10 million people diagnosed as active TB, this TB disease every year, and this doesn't include almost 4 million people underdiagnosed. And also for the pediatrics, this is a top challenging, very difficult to diagnose. And so far in the developing country, the sensitivity for TB diagnosis, active TB diagnosis in children, is still remain around like 30%. So we do have the gold standard. So far is still the culture, the sputum culture. So, but this is a really the long turnaround time procedure, and generally they need 6 to 2 weeks to get the final result. And the most important thing is this test rely on the use of a sputum. And we know not everyone can produce the sputum successfully, especially like children or HIV infected patients. And also in the TB, active TB, the population, we also have like 35% patients, their tuberculosis not located in the pulmonary, they're probably in the brain or neo, and we call those patients as extrapulmonary TB. So the culture based test cannot identify those patients. And we do have the new technology, which is called a Gene Expert. Even it's much faster than the sputum culture, but they still rely on the use of a sputum. So, and also they cannot differentiate the dead bacterial from the live bacterial. And so based on my experience in fighting with this pathogen in the past decade, I summarized the 10 priorities for TB diagnosis. Number 1, is there any way we can develop the rapid method, which is independent of bacterial isolation. And number two, and is there any possibility we can detect the biomarker from the human's blood or urine, and which can better cover the patient with a pulmonary TB and extrapulmonary TB, and also the early detection, which is extremely important for the young children's. And also the quantification method, and so that we can track the treatment efficacy timely, scale up. So how is that possible for us to develop a method similar as the COVID screening and to screen the TB, which will be valuable for the high burden country like China or India, and point of care device. And so this is a lot of need in the other developing country like Indonesia or Philippines, and there are like thousands of islands in those countries, and also the identification of drug-resistant TB. So our group actually is targeting on the different challenging, and we have the three major platforms. And the number one is antigen peptide, the topic I like to present today, and also cell-free DNA, and also the extracellular vesicle study. And but this January, and Nature actually published the Outlook articles and re-emphasized the urgent need for TB diagnosis. And they also listed the 7 promising technologies which can address the TB diagnostic issues in the future. So we're pretty fortunate. 3 of these 7 technology come from our group, including the peptide antigen technology. So let's talk about the peptide, the antigen peptide. So back to the 10 years ago when we just started this project, and we talked with a clinician, the TB clinician, how we can help you guys with the nanotechnology. And they said, well, if you can identify or capture the TB antigen directly from the blood, and you should be able to just diagnose the active TB directly. We thought that this is a really piece of a cake, and as long as there's a protein, and we should be able to detect it. But after 1 year of effort, and we just realized that this is so difficult, and there's 2 reasons. Number one, and those antigen is generally at a very low concentration, and also when they show up in the blood, and they could be quickly bonding with the immunoproteins. So there's a block the detection site in the immunoassay. So and the second challenging is come from the Mycobacterial family. So we know TB is a brother's oldest brother. They also have 190 brothers and sisters. So many of them also secrete the homogeneous antigens, very similar as the target we're trying to capture. And so how to address this? And instead of keep looking at the full length protein, and we start looking at the peptide. And because we think number one, peptide can carry, if our selection is correct, and we can select the best specific peptide, and carrying the amino acid sequencing against the MTB, not other non-tuberculosis mycobacterial. And then number 2 is when we do the digestion. When we do the digestion, you can dissociate bonding between the antigen and also those immunoproteins. So this is really like one stone, two birds, right? So through years, we're actually building up the criteria of how to select the best peptide for this purpose, and also how to building up the pipeline, the mass spec detection pipeline to profile this TB specific peptide. And the first generation actually will focus on the MALDI, and because we believe MALDI is could be high throughput, easy to use, and to facilitate high sensitive detection of MALDI, and we also developed some nanoparticles to enhance the signal. And this is really the first trial for this effort. But afterwards, we realized, oh, actually, a lot of the hospital they actually equipped with LC-MS. So if we want to introduce this technology to the more hospital, to the more customer, and we have to building up the liquid chromatography based method. So then we just generate another the protocol and facilitate this LC mass based the procedure. And when we set up the target, the site of the iron target, and we also building up the criteria how we could recognize this is a true peak come from the antigen peptide. So yeah, that here we building up the several parameters, including the signal to noise ratio, the DOTP and RDOTP, and whenever they fulfill this threshold, and we can confirm this peak come from the TB antigen. But after generating this method, and this still cannot translate them into the clinic because the throughput is quite slow, and then our postdoc actually start searching the alternative solution, and then we focus on the Evosep. So and after the diamond trial, this is really the reformative change, and everything could be processed automatically, and also the turnaround time we can shorten from the 30 minutes per sample to 6 minutes per sample, and this is really the it could be accepted by the clinical doctors. And and after we setting up this method, and we start the clinical validations, and to see if this assay can truly address the clinical challenging in the field. And first, we work with the local hospital in China, and we got a lot of patient samples, but most of them have to be excluded because they cannot fulfill our criteria. But we still have over 300 samples to use, and in this 300, we have 95 culture confirmed TB cases, and we have 102 culture negative but the TB positive patients. This is actually the real ratio between the culture positive and the culture negative patients in the developing country. So in the different group, we also have the pulmonary TB and extrapulmonary TB. So we would like to compare if this assay can offer the similar sensitivity in the culture positive and or culture negative, or pulmonary, or the extrapulmonary. So here the first, because the first evaluation is on the pulmonary and extrapulmonary TB, and because the target we're trying to capture is from blood. So as our hypothesis is, we don't care where this bacteria located, as long as the pathogen is active and they will secrete the antigen into the blood, so we can capture them. So for the pulmonary TB, and we reached the 88% sensitivity, and in the extrapulmonary TB is 90%. So it's a pretty similar. And the next one, we also testing the sensitivity in the culture positive patients and also culture negative patients. So in the culture positive, we reached 92%, and the culture negative is 85%. So this is still very similar. This further validate our hypothesis. And so all this study is on in the adult. And the next one, we actually want to keep challenging this assay using the children, using the pediatric patients, and that is the triple strat for TB diagnosis. And by working with NIH and Harvard Medical School, and we are very lucky to get the large cohort provided from the clinical trial in South Africa. So in this trial, we have the 500 patients, and all these patients actually was recruited when they're only 3 months old, and half of them, they're the HIV positive, but none of them have TB. And then since then, their serum sample got collected every 12 weeks, every 12 weeks. And yeah, then after a few years, some of them developed the TB. So this is a great cohort for us to validate the couple of capability of this assay. Number one, and what's the performance of this assay in young children, because they're like an infant, right? And number two is how early we can detect the active TB from their blood compared to the other testing the options. So so here is the performance of the diagnostic performance, and see that they have like almost 100 the TB confirmed, the TB positive patient, but only 7 of them can be confirmed by the gold standard, which is sputum culture. And in this 7, we capture all of them. And also in the other TB positive patients, which cannot be diagnosed using the gold standard, they have to be identified using the combination of other methods. And in this 65, and we actually reached 89% sensitivity, which is pretty good. And also in the unlikely TB, that means they're just negative for everything. And we use this group as to evaluate the specificity of the assay. We reached 94%. So this blood-based the diagnostic assay can really address the very critical need in the clinic for TB diagnosis. And the number 2, we want to know how early this assay can reach for the patients who has the active TB. So the earliest time point we can reach is 60 weeks before this children's tuberculosis confirmed by the other method. So which is a very promising to further develop the early TB detection platform. And so the next one, we also understand the urgent need in another specific group, which is HIV and TB co-infected patients. So this is like the pathogen brothers. They always exist together. And so there's why this is so urgent, because for all the HIV infected patients, the TB is #1 killer for them. For example, like in the South Africa, 70% of HIV patients died by TB. So this is a very urgent, this is also very important to have a timely detection solutions to cover these 2 pathogens simultaneously. And just by using the same concept from the identifying the MTB antigen peptide, we also identify the HIV antigen peptide, which derived from P24. And by developing this peptide-based detection assay, and we can also reach the earlier detection of HIV compared to the traditional ELISA method. And and also the sensitivity and the specificity was pretty good in the validating the adult and the infant cohort. The most important thing that's how we're going to capture this pathogens change during the treatment for those patients, because we know the patients who has HIV and TB co-infection, generally they will have highly possibly they will have the risk to develop the ARIS. So what is the ARIS? When you have 2 pathogens in the body, they're building up the balance between the 2 pathogens and also your immune system. So when you want to treat one of them, one of the pathogen, you may boost the bacterial load or viral load from the other end. So and causing the dice. So this is a very critical for us to capture the change, the time for this change. So yeah, this method actually the turnaround time for the single patient samples is only 4 hours. So we can definitely provide the timely information to the clinician. So this is an antigen peptide assays, and we actually developed the multiple collaborations throughout the entire world. And it's not only for early detection of TB, they can also distinguish MTB from the other NTM, and also providing the timely result for HIV and TB co-infected patients and many others. So and we also developing the startup company which is called Nanoping, and in the 2020 this technology was recognized by US FDA as a breakthrough technology. And in 2022, the nanopin also received the SBIR award from phase 2 award from the NIH. So the next topic is about the mycobacteria, how we're going to identify or differentiate every single pathogen in this larger family, right? So the different from the blood-based technology, we actually take one step back, and we're not looking at the sputum culture, the solid culture samples. We're actually looking at the early culture solution, that's a liquid samples. And so when we got the culture sample after this culture procedure just started a few days, and we do the very quick protein precipitation, and then we run the digestion for this solution, and then we send the supernatant to the mass spec, and then we got a list, a long list of a peptide. So come from the different antigens. So then we combine those peptides carrying the specific amino acid sequence of interest to identify, to confirm, and which pathogen or which subspecies that the patient could be infected. And the principle is pretty straightforward, but we're actually building up the very comprehensive software or algorithm to guarantee that this combination is accurate. And so we call this a one-step classification of mycobacterial from the species to substrain in the one spectrum. And so here is some practice. And so in the US, generally the doctor like to target on the MTB, abscesses, kansasi, and the Mycobacterium and intracellular. There's 5 major species, and we're just using up to 15 peptides, and we can address this issue in the time minor. And also, if you go to another country, they're pretty focused on the mycobacterial abscesses. But under the abscesses, there's like 22 different subspecies, and some species is drug resistant, and some species is drug sensitive. So we also have the bunch of peptide to differentiate. There's 22 subspecies, and this is all we like to present today. And this is funding, all the funding supporting our research. And here is our group. Thank you so much.